I'm Ilan Gronau. I'm from the Herzliya Interdisciplinary Center in Israel, which most of you, if I think probably 90% of you haven't even heard before. So it's a really nice place, as you can see here. This is a view from my window. So I'm going to do two promotions. I don't know if those are approved of this, but I'm the first one giving a tutorial, so I guess I can set the standard. So first of all, I'm going to promote my lab. So if you want to come and visit, probably not during the summer, but during the winter or spring, it's a great place, and I'd be happy to host you. And another thing that I'm going to only, wait a minute, clicker problems. Okay. No. Good old school. I'm going to promote GFOX, which is a method I'm going to talk a bit about during my tutorial. So I try to keep it as general as possible, but you're going to see kind of a few mentions of this method for demography inference. So what I'm going to try to do is try to go fairly slow about what Bayesian demography inference is, and there are quite a few methods that kind of use this approach to study population history. And in the second part of the tutorial, I'm going to talk about a problem that I've been thinking about now for a few years, and we're starting now to get tractions on solutions, and I think it's a really neat problem that I don't think has been given the kind of appropriate attention in this field. So what I do in general as a kind of computational population geneticist, and those of you who've been to the retreat kind of will be familiar with some of these introductory slides, is that we try to understand how population history and evolution in general influence genome sequence variation. And one of the reasons we're interested in this is so that we can take all of this great data that's coming out of sequencers so that we can make cool inferences about population history and evolution. And there are kind of two main components to this, and I'm going to talk only about the first one, which is kind of reconstructing a demographic history of population. There's another, great application to studying natural selection and evolution that I'm going to completely ignore during this tutorial. So when I'm going to talk about a demographic model, it's going to be something like this. So different people have kind of different notion of what a model is, but this is something that I find quite useful. So I'm going to describe a demographic model as a tree where my leaves are kind of populations that I can see in present day. These are the populations that I'm analyzing where I'm actually sampling my genomes, and they're connected via some kind of conceptual population tree. But because this is, populations don't live in complete isolation, then I'm going to allow kind of these points of contact, horizontal points of contact between these populations, which I'm going to call migration bands. So a demographic model for me is this kind of tree where each internal node is going to be associated with kind of a time at which that split event happened, and each branch of the population, phylogeny, I'm going to associate it with kind of a typical effective size that's going to kind of represent the genetic diversity during that period of time, and I'm going to have kind of these rates of gene flow between populations. So this is what I'm going to try to learn from my genomic data, and this field is advanced in many kind of different directions, and the kind of particular angle that we're going to take, and that's very useful in these Bayesian methods, is to kind of think about the genetic genealogy in the middle. So what is a genetic genealogy? In order to understand that, we have to first understand what a genealogy looks like. So genealogy is pretty much what you would think it is. If you have like a focal individual here, this is, say, an individual for which you have a genome sequence, then you can think of this individual's ancestry going back in time as this tree. You have 2 parents, 4 grandparents, 8 grandparents, etc. And at a certain point in time, these branches start coalescing because you have ancestors that you can kind of reach in different ways. So this is not strictly a tree, it's kind of a graph where each node has two parents, but some nodes might have more than one relevant children in this graph. So why are genealogies very useful for reconstructing demography and population history? So you could see that even if you kind of have the genealogy of a single individual, because it traces through many individuals in the ancestral populations, then just by looking at the genealogy, you can kind of see certain demographic events that influence the ancestral population, like a decrease in population size will be observed by kind of an increased rate of these coalescent events, because you have few individuals that can act as ancestors, so you're going to see an increased rate of coalescence. So this is a very basic kind of concept in population genetics to try to map rates of coalescence with population size. So this is what you can do with a single individual, and if you have multiple individuals from different populations, then you can obviously learn kind of their history of their population sizes, but you can also learn a lot about the relationships between these populations. For instance, here, by looking at this kind of combined genealogy of 3 individuals, you can see that these two populations from which these 2 individuals are taken are more closely related than they are to this third populations, and you can even try to estimate their times of these splits by looking at, again, the distribution of these coalescent events now between parts of the genealogies of different individuals from different populations. And where do we get these kind of migration rates from? So if we assume that these populations do not operate in isolation, then you're going to have kind of these cases where lineages cross through population boundaries, and that's what gives you information about gene flow and migration. Right? So this is kind of the basic idea of how we can get, from genealogies information about population history, and that's pretty much an obvious idea. I think the only thing that's not obvious here that's really neat is that you don't have to have many individuals, right? Because each individual carries a lot of information about their entire ancestry. But the main question here is, how do we get information about these genealogies from genome sequence data, right? We can never actually see these genealogies. So that's the second part, and that's, again, introduces some really cool models. So say we have these 3 individuals, and you can see now their population history in this, and the genealogy kind of in very faintly in the background. And now we have their genome, so we don't actually see this. So where can we find kind of hidden information about their kind of combined genealogy? So say we focus on a certain site in their alignment, in their genome alignment, then every site along the genome is kind of, you can kind of think about the parts of the genealogy that relate to that site. And these are kind of the paths, the lineages going back in time where that site has been kind of inherited from a common ancestor. So if you go from a certain individual back in time, then in every site you have one lineage of inheritance, right? You never split into 2 parents. You kind of, every site came from a single one of the 2 parents for each of these ancestral individuals. So the way it looks when you kind of overlay it on top of the big genealogy is that you kind of get a subtree of this big genealogy construct. And that's what you happen when you kind of, that's if you look only at a certain site, and there are obvious connections between the site patterns. So if you see no mutation here, if it's kind of site without no polymorphism, then you don't really get information about the genealogy. But if you have a mutation, then you can think about, the mutation has to sit on one of these branches, and you do get some very weak information about that genealogy. So single sites are not very useful in general in getting very good genealogical information, but when you kind of expand that window a bit, you still get, you still get the same genealogy, so that same genealogy is going to kind of influence not just a single site, but typically, a very short window around that site. But then at certain points here indicated by these dashed lines, you encounter a combination event that flips kind of the local tree. So to the right of this dashed line, you might get a different tree due to a combination event at this point that kind of switched from mother to father, and you get a different recombination event at this point that gives you a different tree tracing back through the same big genealogy. So the basic idea, if you kind of further zoom out, as you go along the genome, you have these local trees, or what I'm going to call local genealogies, that flip around, and hopefully you get a good enough characterization of the big genealogy that gives you information about population history. So the information is somehow hidden there. It's very, very noisy because the sequence patterns you actually observe provide you with very weak information about the actual genealogy at that point. But there is some information, and the whole kind of trick behind doing population phylogenomics is being able to kind of read these patterns and interpret them in terms of population history. So one final thing is, again, so this structure, this tree that evolves along the sequence is called the ancestral recombination graph. I think that it's going to be mentioned in several of the computational population geneticists giving talks, so it's a very useful term to keep in mind, and it's something that I always kind of have in my head when I try to think about these problems, is what does the R really look like? How can I actually use it? So how does that relate to demography inference? So you would want to actually use the complete R to do demography inference, but that turns out to be very complicated. It's not impossible. I think down the road people might be able to make it work. But one way to make it work that's significantly simpler is to avoid looking at kind of the long stretches of sequences and kind of think of your genome as in these very short windows that are spread apart. And the way that that really helps is that now you can assume that your data is kind of governed by a series of independent local genealogies that are just trees, and you hope to get enough information about these trees to give you information about the population history. So you don't have to model kind of the very intricate process of recombination. You just assume that each locus that you're analyzing, there are no recombination events there, so there's just a single tree, and there are sufficient recombination events between loci so that you can assume the trees are independent. So obviously, to make kind of these ideas work, you have to make sure that these kind of slightly false assumptions you're making are not going to hurt you, and that's pretty well established. So now how do you use it to do demography inference? It still doesn't solve the problem. It's still a quite complicated problem, but as I'm going to show kind of in the next 5 minutes, it at least makes it somewhat tractable. So the idea, again, is that in all of these kind of Bayesian demography inference methods is that you assume kind of a known structure for your population history, and you try to use information about these local trees along the genome to make inference about the demographic parameters, which are going to be the split times, population sizes along branches, and migration rates. And we're going to do that using Markov chain Monte Carlo sampling method. So because this is the first tutorial, then I'm going to try to describe what this Markov chain Monte Carlo sampling method looked like in this context. So if you're not at all kind of familiar with MCMC, this will hopefully be a reasonable introduction to what MCMC does. So again, this is kind of our starting point. So if you want to do demography inference, somebody gives us these genomes, and we choose these windows wisely or not wisely. We don't actually have to be very wise in choosing them. It's actually almost better to do it at random than to try to find the right places, but wherever we have good annotations, we want to kind of fixate them around places that we think are not strongly influenced by natural selection, because the evolutionary models we're going to use are going to assume neutrality. I'm not going to talk too much about that, maybe a bit more in my research talk next week. So that's my data, and this is not part of my data, but it's part of kind of my assumption. So I'm going to assume that I know what the population phylogeny, the structure is, like which populations are more closely related to the others, and I'm going to add on top of that scenarios, possible scenarios of gene flow. So in the second part of this tutorial, I'm going to talk a bit about, okay, so how do we figure out this part? That's the tough problem. But say we can make some assumptions about this structure. What we're actually interested in are the model parameters associated with this model, the split times of these internal nodes. These are these taus. The thetas are kind of the size parameters of the branches, and these m's are the migration rates of these green arrows going between populations. So our objective in this MCMC sampling procedure will be to sample values of these model parameters according to what we would hope would be a posterior probability distribution. So it will be the probability, given some reasonable probabilistic model, we'd want to sample values of these thetas given the data, which is what we have at hand, and the model M, which is something that we assume. As we assume we know. So that's our objective, but it's quite complicated even then, because we can't actually compute any of these probabilities. We can't compute the joint probability of the data and the model parameters given M, or any constellation here, because we're missing something. So for this, we can make approximations. So some methods do this by making some approximations about this probability. What we're going to do is going to make the problem more complicated so that it becomes simpler by adding additional kind of variables that we don't have to begin with. These are these genealogies, the genealogy kind of associated with each locus. These are things that I don't observe. They're hidden variables in my model, but once I introduce them, the nice thing is that the problem actually becomes tractable. I can at least compute now these joint probabilities, the probability of the data, the genealogy is a genealogy for each locus. This is what this G stands for, and my model parameters. So I'll get into this kind of equation in a bit, but just kind of understand what the components here are. So again, X is the data that I actually have. M is the model that I'm assuming. Theta are kind of my target. These are the things that I actually want to get out of this method. And G, these genealogies are something that my method is going to have to kind of internally hold, but I don't care about it. As somebody who just wants to understand population history, I don't really care about what these genealogies are, as long as they help me get good parameter values. So what does this joint probability distribution looks like? Maybe I'll do this here, it'll be easier. Then first of all, because it's kind of a fully Bayesian way of thinking, then I'm going to have priors to my parameters of interest. So these are somewhat arbitrary. There are certain distributions that are used for kind of ease of use, but hopefully I hope that my posterior distribution is not going to be significantly affected by my prior. So if I have enough data, if I have enough terms here, this term kind of doesn't really matter. That's the idea. So I have this one term for the prior, and then I have a term for each locus that I'm analyzing. This is what this product stands for. And in each locus, I have a probability of the genealogy given the model and the current set of parameters that I'm assuming, and a probability of the sequence data given the genealogy. And the nice thing about introducing these genealogies is that given the genealogy, the data and the model become independent. So the genealogy kind of is a really good middleman to use to make, again, this computation practical. So I have expressions for pretty much all of these. For this, I'm going to have to use kind of the Felsenstein pruning algorithm, but it's a very quick algorithm. So it's not really closed form, but it's something that computer program can compute really very quickly. So I have now, again, by adding these genealogies, I have a framework for actually computing scores for the instances that I'm going to use, these genealogies and the model parameters. So what does MCMC look like? I start at some arbitrary point. I just choose a genealogy for each locus and values for all my model parameters. And then I iteratively kind of update my current state of the universe by choosing either a parameter to kind of perturb or a genealogy to change. So say I want to change this genealogy, these changes are going to be typically going to be kind of small local changes because I don't want to disturb this likelihood, this probability, this joint probability too much. So I'm going to say take one branch and try to kind of reattach it in a different place. I'm going to recompute my likelihood, and I'm going to see what happens. So if the likelihood grows, so again, they're kind of somewhat not so sophisticated but intricate kind of decisions to make here. But in general, I just compute the value before my proposal and after my proposal. And based on these two values, I make a pretty easy decision whether I kind of choose to accept this proposal, and now this is going to be my next sample, or reject it and kind of stay. My next sample is going to be identical to the previous one. Right? So it's a pretty, conceptually it's a pretty simple algorithm. It has these proposal, acceptance, rejection steps, but you have to kind of design it reasonably well so that it can actually, traverse a very, very large and complex space and hopefully get you to the parameter values you're interested in. So again, I think maybe I don't want to belabor this too much. So the idea again is that in each sample you get an instance of all of your genealogies and parameter values. The first few, I'm saying few, but it's probably thousands, tens of thousands of samples you're going to have to throw away because you started at some arbitrary point. But then at some point you're going to assume, you're kind of converging, and then again you throw a lot of your samples away because two consecutive samples are going to be very highly correlated. You make these very small differences. So if you want to reduce the level of correlation, you just use longer scales. So that's what makes MCMC really typically very slow is again this fact. You have a series of pretty complex computations and you have to let it run for a long time. But the nice thing is that if you do wait for enough time, you're guaranteed that your samples, and what we care about here is only the model parameters, are going to be distributed. You're going to get distribution that is going to converge to the posterior distribution. So it's here, if we throw away the genealogies, it's exactly what we wanted. Model parameters distributed according to the posterior, given your data and the model that you assume. So here I had I felt like I had to give sufficient credit to the people who kind of introduced this to our lives. Rasmus Nielsen, John Wakely, and Jody Hay through IM, and later MCMC Cole and BPNP by Zheng Yang and colleagues. And I arrived pretty late to the game and with GFOX, but I think the nice thing about GFOX is that we really optimized it for doing this population level inference with multiple populations, a lot of migration banks, fairly complex models, and a lot of loci. So we typically run these analyses with tens of thousands of loci genome-wide. And we've optimized the program really nicely to be, so that you could run it in reasonable time. So just to kind of show you that this actually works, I thought I'd do a demonstration out on real data. I'm going to talk a bit about real data analysis during my research talk, but on a limited simulation experiment that I think exemplifies, first of all, that, hopefully it will demonstrate that this works, but where you can kind of get a lot of the complications that people actually encounter when they do real analysis. So in this kind of toy experiment, we have this very simple model with 3 populations. There is a constant population size throughout this entire history, and these kind of 2 divergence times. And all these numbers you're going to see are actually scaled up by 10,000. So they're 10,000 times smaller, but it's just, I think, easier to see these numbers. And I'm going to have some gene flow here for the sake of the example between this kind of outgroup population and population B. So we simulated data according to this fairly simple model using 2 individuals, 2 diploid individuals in each sample. So it's actually 4 samples because each diploid actually, we modeled kind of the 2 copies, and we don't even have to know the phase. It's a nice feature of GFOX. And we simulated 2,000 loci, which is typically a lot less than we'd actually analyze from genome sequence data. But I did this just so that I can, generate quick enough results. And again, this is a cleaner model because I'm going to actually simulate based on the model that I'm assuming. So it's a very toyish example, but just to show you that the signal is actually there and can actually be captured. So these are going to be 2,000 loci, 1 kilobase long each. So this is the data that we simulated, and the first thing you want to see is, do you actually get good parameter estimates if you assume the right model, if you assume, the correct model. So we assume we know the structure of the population tree, and we added a bunch of migration bands. So not only this migration band here, there are actually 6 migration bands here, all pairs from all kind of leaves. You can actually have even migration from ancestral populations in the model. So what do we get when we actually run this inference? We get pretty good results. You see all the population sizes are nearly, nearly identical to the simulated ones. These are kind of the 95% credible intervals. They're decent. The 2 divergence times, this lower one is really spot on. This upper one seems to be slightly underestimated, which could be due to the prior. So the priors here are, I typically use non-informative priors, but here, I think kind of by accident, I assumed a very deep divergence here and didn't reset my prior to kind of say that my prior knowledge is that this divergence time is deep. So this slight underestimation is probably due to my prior knowledge and the fact that I have only 2,000 loci. So if we push it to 10,000, I'd expect this number to go up and be pretty close to the simulated value. So these are the 2 divergence times and the population sizes. What about migration rates? So you see this migration rate for the band between C and B, where we actually simulated gene flow, and you get a pretty accurate estimate there. And for all the other migration bands, you get, so you get pretty wide credible intervals here, but they all go all the way down to 0. So I can't reject the hypothesis. One way to think about it, it's not statistically accurate, but you could interpret it as not being able to confidently reject the hypothesis of a 0 migration. But you do see, for instance, this odd thing where we do seem to be estimating kind of significant levels of gene flow between these 2 sister populations. And this is something that often happens when you do this analysis, because it's pretty difficult if you have 2 sister populations. It's pretty difficult, and say you even saw the genealogies, it's pretty difficult to say when divergence starts and gene flow, where divergence starts and then you start seeing gene flow, or whether it's just a lower divergence event. And you actually see it if you look at the traces. You actually see the method kind of struggling there. So I show here the 3 relevant parameters: the deeper tau, the tau of the root, which is pretty stable. First of all, you also see the burn-in, which you don't see because it's very, very quick. It's within, I think, 1,000 iterations in these simulations kind of reach the level that you'd expect. And here you kind of see this this is sampling that I assume is already kind of pretty much according to the posterior very early on. So this tau root is very stable, but this lower tau seems to be kind of fluctuating at this point, and it seems to be related again. So you can kind of assume that the divergence between A and B is deeper if you add migration between A and B into the model. So the model kind of just shows you that the model explores that possibility. It seems to be kind of transient. I guess if you let it sample long enough, it will again transiently visit that space of the parameters. So it gives you a pretty nice idea also about your certainties and uncertainties. We can't really truly be uncertain about the divergence here once we add a possible band, kind of a gene flow between A and B. So we get pretty nice estimates. But that's very clean. What happens if we assume the wrong model, which is something that we probably often do when we actually analyze data, because we don't always know what the model is. For instance, we might not know that there is gene flow here, so we can run the nice thing about methods like GFOX or the other Bayesian demography inference methods. They're reasonably quick so that you can actually run on multiple models and try to see what you get. So if you run without migration, you get kind of the expected things. You get a reduced divergence between C and the others. Because you're not modeling gene flow between C and B, then the model kind of has to pull this divergence time down. And you get also some differences in the population sizes that, again, are due to the fact that you're not modeling gene flow. So you get something different if you change your assumption, which is not very surprising. And you get something even more different if you actually flip the topology of the tree. So if you assume that it's not A and B that are sisters, but A and C, then you get all sorts of weird things. For instance, you get these two divergence times because now A and C are sisters and A and C are quite divergent here, then this gets pulled back up and your two divergence times are going to be kind of stuck together, and you're going to have a lot of gene flow between A and B, right? That were originally you simulated them as sister populations, and now they're kind of more distantly apart. So the model is going to model it using gene flow. So what I'm trying to get at here is that when you actually do data analysis, you don't know what the correct model and what the incorrect models are, but you can run the model, several times, and then all you have to do, at least with a lot of the current methods, is look at the parameter values and try to kind of read within the values which model might be more correct. So we've turned to be pretty good at this, reading in the tea leaves of the parameter values, and we've been pretty successful with that, but it's not really the way that you'd want to go. You'd want to actually have a nice way to be able to run different models and be able to say, okay, I can score this model, give it a certain score based on the likelihood, this model give it a different score, and this model give it a different score, and then say, okay, my model of choice is this one because it has the highest score. And this is something that's not really implemented very well in any of these Bayesian demography inference methods. So IAM does have kind of a feature about that, but I'm going to argue that it's not generalist enough. It's not going to work, for instance, I think, in this type of situation. What we want, what we strive for, and this is a problem I've been thinking about, I think, now for 4 years, is to have a general method that will allow us to actually score very different models if we want, and get reliable scores that we can actually use in a publication, because this is what we all want. We want to get our genome data, run different models, we have time, but then we want to be able to say this is the one that came on top. And we can't currently do that. So yeah, I have, how much time do I have? 10 minutes. Okay, so I'll try to give you an idea of kind of our thinking about this and what we think can be done. And this is all new. This is stuff that I've never actually presented before, so it's actually kind of a research component in this tutorial, but I'm going to try to really address the basics of what I think, what are the nice ideas that can be used here. So the main thing, and this is something that is not new, that's been around for a while, is to try to use importance sampling in some sense to estimate these values. And the idea behind importance sampling is, if you're not familiar with it in the general sense, is that say you have a function f, and you're interested in computing the expectation of f, giving a complex distribution. This is your p here that has these two modes, but you can't sample under p, so it's very difficult for you to estimate this expected value. What you can do is sample according to a simpler distribution q that is somewhat similar to p, or at least what you have to make sure of is that it support the values that it gives non-zero probabilities contain the values that p gives non-zero probabilities, as you see here. So if you have this scenario, then you can actually express, I'm not going to go through this in too much detail, but you can actually express this expected value that you want as an expected value under Q, but then you can use this kind of very simple reweighting approach to kind of weight the different values of f, again, proportionally based on the ratio between P and Q. So this is a pretty simple idea, and it works reasonably well as long as P and Q are close enough. And it's going to work better if P and Q are more similar to each other. So this is an idea that we're going to try and utilize. So where does importance sampling come, in our approach? So I'm trying to think about it very generally now. So we have this model, which is this, our general assumptions about models of evolution plus this kind of tree structure. And we have our data, which we know are these unlinked loci. And another part of our kind of probabilistic machine are these hidden variables that we have to add. So there are these, here I'm going to kind of lump together the model parameters and the genealogies, because what I want to score is not the model parameters that I'm inferring. I actually want to score my structural assumptions. The structure of the phylogeny, the migration bands that I have there, that's what I want to kind of score eventually. So the parameters and genealogies are kind of lumped together in this notion of hidden variables. And the idea is that I worked very hard to produce samples of these hidden variables, right, with my MCMC, but I'm not really using them that much. I'm using them to get a posterior on my model parameters, but I'm not using them. But what I want to do is use them to also score my assumptions. And I'm going to want to use importance sampling to do this. So this is a distribution. So again, these y's, by virtue of using MCMC, I'm guaranteed that at some point these y's are going to be distributed at least closely to the posterior probability of y given x and m. So this is a distribution I actually have access to. In this term, that's my q. But I want to use this q to compute a more complex function that I don't have access to. So this is a slide with kind of a series of equations that I don't have time to get into, but hopefully you'll believe me that you can actually express the inverse of your likelihood as an expected value, given the posterior. So this is my Q. This is a distribution I actually have access to by using MCMC. And all I have to do is record again these probability of the data given my hidden values in the model, which is only going to be my genealogy. So these are actually values that I record during my MCMC. I just have to print them out and then compute these expected values. So this is not a new idea. So this is something it's often called the harmonic mean estimator, because again it looks like a harmonic mean. The inverse of your value is the average of inverses of relevant values of interest, right? So this harmonic mean is, seems like it's, it conceptually seems to work, but we have a big problem with that, its variance is very big. So while we are guaranteed that if we have enough samples, this is going to converge to the right value, the variance is just too big, and we never actually have enough values for it to converge. So what I think I'll end with is just an example to show the problem and just to give you a peek of the ideas that I think can actually hopefully get this kind of field forward. So we've done, again, a bunch of simulations similar to what I've shown before, but in this case what we've done is we simulated different data sets and kind of varied this lower divergence time to see, and in these different cases we wanted to ask whether we could tell a very simple thing that you'd expect would be easy. Could we tell whether this data came from a model where A and B were closely related, or whether it came from a model where A and C were closely related? And this is without gene flow, so it's just being able to tell what the structure of the phylogeny is using the MCMC. So you could probably do it easily in this case without actually using the MCMC sample. So it's a toy example, but I wanted to show you that where kind of the standard approach actually breaks. So we looked at this as a function of what happens when you play with this tau value here. So this is what you see here. These are different values for this tau that starts at very low, 0.1, and goes almost all the way to the top to 2.9 up here, right? And you can see that when it's very far, when this tau is very low, so when there is a strong signal about relatedness of A and B, we do pretty well. So these are kind of the values. We want them to be high, and these are kind of bootstrap estimates of our uncertainty. So you can see that already kind of the indication of very high variance, but you see it does reasonably well, but then it pretty much crashes when this tau becomes 2, which is pretty far from here. So we still want to be able, there obviously is signal about the topology of the population phylogeny, but we just can't recover it using this method. So what I'm going to end with, is just a really small sneak peek to kind of the ideas that we're thinking about to solve this problem. Is that the problem with that harmonic mean estimator is that you estimated a separate value for each model, and you never really used the fact that these models might be very similar. And that's actually what happens when you take this tau and push it all the way up here. The models, the different topologies become actually more similar because, again, the genealogies that you generate under the different models become more similar. So in a sense, because the models are more similar, you'd want the problem becomes tougher, but you'd want to use their similarity to make your estimates more robust. So our idea of solving this is by introducing what we call a reference model that we can use to compare each of these models directly to. So first, these estimates, original estimates, are kind of reference-free. They don't care about any context. But by actually adding context and saying, okay, I don't want to compute the likelihood of these two models, but I want to compute what's called the Bayes factor, which is just a relative likelihood of a model compared to another model, but I'm going to compute Bayes factors relative to the same reference. I'm going to use these, and if I can estimate these kind of more accurately than I can estimate the original likelihood, then I can get better estimation, better comparison between these two models. So I'm not going to be able to get to the equations. They're very similar, but not quite, and there's a neat idea here that's kind of hidden in the details, and that's something that we've had to kind of pan out in the past year or so, as when can you actually do this, what are good reference models. So that's kind of part of the research component of this tutorial that I'm not going to get into. But just to kind of finish on a positive note, we kind of redid this whole experiment, but now did these comparisons using this reference, and it works pretty well, as you can see here. So it's not that I removed the error bars. The error bars are pretty much kind of out of scale of the actual values that I'm estimating. So we can reach almost up to 2.9 here and get very good and confident call for the correct model, which is what we want to get. And we get this added kind of sensitivity down here because we use the reference model and not just do it without any reference. So just to conclude what you saw in the past 45, 50 minutes, we talked about Bayesian demography inference and why it's really useful, sometimes at least, to think about these genealogies in the middle, and that really helps. Again, there are a suite of methods that actually use this to solve these cool problems. And we talked about, I think, what's one of the major deficiencies currently in the field is the lack of good methods to actually score different assumptions that we make on the model. And this is very important because otherwise we end up saying, okay, we assume this, and then we get these divergence times and sizes and migration rates, but the assumptions are usually very, very fuzzy. And all of this, again, we hope to make a lot of this progress and kind of implement it in GFOX. So if you're interested, so kind of keep track and see what's going to be new. Hopefully we're going to have something out in the next few months. And that's it. Thank you.